Good morning, everyone. Dr. Bijal Prajapati, Associate Professor from Parul Institute of Pharmacy, Parul University, welcome you all on webinar for the Pharmaceutical Production Management and Technology. Will be presented by Mr. Jayesh Don, Research Scientist, APL. I would like to introduce him. Mr. Jayesh Don has done his B Farm from North Maharashtra University and M Farm from Vinayaka Mission University in 2011. He is a technically astute and diligent professional with more than 8.5 years of industrial experience. He possesses of the knowledge of manufacturing for various drugs, research and development, strategic planning, technical troubleshooting, and workforce supervision. He has core competencies in formulation development, technology transfer, team management, and documentation and data compilation. He is currently working with APL Unit 7 and with his profile on the suggestion of batch size and equipment management according to the occupancy, capacity, and regulatory requirement for ANDA, dossier extension, site transfer, and commercial launch products. He is coordinating, planning, and reviewing the TTD of transferring location and also responsible for preparation and reviewing of documents for scale-up, trial batches, and validation of batches for the received as per the location requirement. So with this short introduction, I would like to invite Mr. Jayesh Don to present on pharmaceutical production management and technology. Over to you, sir.
Yes, thank you for a good introduction, ma'am. So, good morning to all of you. So, I would like to thank for giving me the opportunity to have a speech regarding for the pharmaceutical production management and technology. So, as per my brief introduction has been given from the ma'am. So, I would like to just give a small introductions regarding what I am. So, myself, Jayesh Down. So, as a technical student and delegate professional, spreading as an research scientist in APL unit that is located in Unit 7. So shall we start for the presentation?
Yes, yes, please.
Yes, thank you. So today is pharmaceutical production management. So we will go what is actually the pharmaceutical production management, because all of the aspirants who are now online will be aware of the GMP. But what exactly GMP follows in the pharmaceutical production, I will just explore it. So the GMP is nothing but the good manufacturing practices that are a part of quality assurance that ensures that the drugs are consistently produced and controlled in such a way to meet the quality standards appropriate to the identity use and required by the marketing authorizations. So part of the health products and the food branch expectorant, this product conduct to the inspections of establishment that are involved in the activities covered by the established licensing framework. So these inspections are conducted to verify the compliance with the GMP part. Okay. So we will go for the applications, what the GMP applications are. So it's generally the structure of the organization to build the quality system to meet the requirement, establish the policies, objectives, and plan, review the system, analyze the data for the trends, conduct the internal audits, risk assessments, and promote the improvement. So the pharmaceutical industry is a major global economic force which increasingly relies on the safety, efficiency, production, and a technical advanced product. So the environment challenge the facilities professional who is charged to plan, to design, to construct, to validate, and to operate the complex manufacturing factories that meet the world-class pharmaceutical operating standards. Coming to the next slide. Slide number 2. Next. Okay. So what are the considered factors that are used for the selection for the plant location? The first one is the availability of the raw materials, that is nearness to the potential market. What does it mean by potential market? The potential market is that the volume, what exactly the market requires. On the basis of that volume, we have to achieve the availability of the raw materials. Third one is the locations should be near to the source of operating power, supply of labor, transport and communication facilities, suitability of the land and climate, integrations with the other group of companies, availability of the housings and other amenities and services. So these are the points which makes the hierarchy for the GMP for the good pharmaceutical management as well as the production planning. Okay. For the next slide, plant layout. Once the decision about the location of the plant has been taken, the next important problem before the management is to plan the suitable layout for the plant. So plant layout is the method of allocating the machines and equipment, various production process and other necessary services that are involved in the information process of the product with the available space of the factory. So production planning layout is mainly done in a such a way that a step by step we have to follow the individual steps. For example, for starting from the raw material, so what is the next step is the receiving of the material, then sending of the material, then warehouse is there. Okay, so these are the layout diagram in which pictorial explanation can be given in easy way. So first we have to receive the after receiving of the raw material, it is to be gone to the warehouse. From warehouse, there are two components are there for the drug components and the packing materials. So once the drug has been reached and packing material has been reached, there is a quality control inspections to be done whether the component and the packing materials which are being intended are to be rejected or to be kept. They have been analyzed. After analysis, the rejection will be thrown and the good one will be kept. So weighing and dispensing will be done for the next step. So after approval of the material, so manufacturing process will be start. Then in process storage, then after the process storage has been done, there will be in quality inspections. After inspection, the compliance will be given and the packing and labeling of the product will be done. So it indicates that the initiation of the raw material from the starting source till the finished product, that is the packing. Once it is completed of the packing, then it has to be done quarantine. Quarantine means it's a storage only where the drug has been kept in a suitable environmental and rheumatic conditions. So once the drug has been kept in environmental conditions, it has to be released based on the production storage, and afterward is the shipment. So this is the building layout design and the construction of the plant layout. So we will go for the detailing about the building layout design and the construction. The first step in the construction of the building is to make out. So we have to keep many points in the mind. So building layout destruction. So depending upon the total area of land, percentage of the area that has to be covered under the local laws, which type of dosage form we have to form. So depending upon the dosage form, we have to lay out this plant. So type of the equipments. Now, depending on the type of equipment, size, capacity, so area as well as the allocation of the land has to be done. So requirement of separate building for the hazardous. So you know that different types of dosage forms are available depending on the beta lactam penicillins, oral solid dosage. Then one is that the non-epileptic, aseptic manning, so parenteral, ophthalmic, this type of dosage forms are there. So for that we have to give a different facilities and different services. Okay, you're getting. Now we will come that types of manufacturing layout. So first one is the product line or line layout. So in these machines, equipments are arranged in one line depending upon the sequence of operations that are required for the production. Okay. Second one is the process of functional layout. In this type of layout, machines of similar types are arranged together at one place. Okay. Next one is the fixed position or location layout. This fixed position or location layout, this is the major product being produced in a fixed at one location, one equipment, same labor and same component that are to be moved to that locations. So these are facilities are brought and arranged around one work center. Next one is the combined or group layout. So combined and group layout, in this manufacturing units may require all three types of process, namely intermediate process, continuous process and prospective repetitive process. So these are the types of manufacturing layout. Now we will go for the facilities and the services. Okay. So facilities and services which mainly includes water, that is a potable water, purified water or distilled water. Steam, which is to be required, that is to be heated at a more than 100 degrees Celsius to get converted into steam form. Gases, that are the air, natural gas, nitrogen gas and carbon dioxide. These gases have their role when something is explosion and that has to be taken. So for that reasons we are using the gases. Electricity and lighting, HVAC system, that is the heat and cool air condition, ventilation, filtration, humidity, cleaning, sanitation and maintenance, fire, health and safety, drainage system and ATP, that is the influent treatment plant, security and control, and transportations. These are the facility services to be given when we are deciding for the layout. Now we will explain what is the next slide. Coming to the next slide. Yes, this completed. Next slide. Okay. Now in this type, there are number of facilities are there. So I have just coming to the point. What are the main equipments facilities that are required for the plant layout? So mainly we will explain about the HVAC system. So HVAC system is very important because it mainly gives the information for the airflow distribution as well as the control, filtration, air recirculation, supply airflow in sufficient volume, cleanliness to the support to the cleanliness rating to the room. Okay, introducing the air in a manner to prevent the stagnant areas where the particles could accumulate. So HVAC system, as we know, in a degree or in a post graduation course, we will learn about the HVAC system. So we will know about the different class of HVAC system for the different area. So depending upon the different class of area, what do you mean by different class of area? For the dispensing and weighing, there is a 100 class where the air can be circulated, and to prevent the suffocation like activity where dusting of raw materials can be happen. So to inhibit or to prevent this kind of suffocations like activity during the dusting of raw materials, for that reason HVAC systems will suck the air and it will just polluted free will be given for the labor or the compounding of system. So these are the main thing for the HVAC system. So when pictorial representations are given, where I have rated two types of pictorial for HVAC system. First figure represent that different areas having a different HVAC system. Okay, and second figure that indicates that a working culture where the peoples can have a good supply of air during the from the HVAC system. Okay, so whatever the finished products should be suitably designed and constructed to facilitate the good sanitations. Now we will go for the next slide where it includes the ETP. ETP is nothing but the effluent treatment plant. Whatever the waste for during from the initiation of the raw materials and during the manufacturing and operations, whatever the waste rejections are to be collected in a separate area where we can treat this all the wastage in a such a way that we can make a slurry sludge, and we can heat in a such a way at a high temperature more than 800 degrees Celsius to convert that sludge digestions into a powder. Okay, so this helps to inhibit or to prevent the microbial or environmental contaminations. For that reasons, ETP plant is must and it should be with a part of the plant layout only. Okay, so the ETP plants are used widely in the pharmaceutical industry to remove the effluents from the bulk drugs. So during the manufacturing process of the drugs, varied influents and contaminants that are produced, the effluent treatment plants are used to remove the high amount of organic debris, dirt, grit, pollution, toxic, non-toxic materials, polymers, etc. from the drugs and other medicated stuff. So the pictorial representation is given for the pre-treatment. For whatever the overflow that is has been tried, whatever the air dress has to become. So primary aeration means what? We will dissolve all the materials in a slurry form. Okay. After that slurry, it will be evaporated to the treatment plant and it will be dried in a suitable finishing product, and the methane and the gas will be evaporated and the fine drug will be present. So this is nothing but the ATP. It's the main source of plant layout. Okay. Now we will go for the departments in the pharmaceutical industries. Next slide. So after having a different types of layouts, whatever the layout has been finalized, so we will go for the section wise pharmaceutical development industry, that what kind of department have the different stages. So first one is the central warehouse. So in simple words, we can say that central warehouse is nothing but a storage room. So this is further divided into the two types, that is a raw material warehouse and second one is the finished good warehouse. So raw material warehouse mainly includes the active ingredients, that is the API. Next one is the excipients, and third one is the packaging materials, which includes in the raw materials. And next one is that finished goods warehouse. That is nothing but the generally the nearer to the outlet of the company for the easy and the efficient transportations. Once the drug has been converted into dosage form, it will be kept in a finished good warehouse, that is in quarantine area, and it will be nearer to the outlet where the dispatch of the material to be taken place. Now we will go for the production department. So in production department, there will be a number of department will be there. First one, we will go for the liquid department. So I will elaborate what is the liquid department. So there are three types of liquid preparations. One is solution, suspension, and emulsion. So for solutions, the equipment for the solution preparations mainly includes dispensing booth, preparation and manufacturing tank, that is the SS, steam jacketed. Third one will be storage tank, bottle washer, filter press, filling machine, capping of the bottle, and last one will be the bottle labeler, that is the automatic, semi-automatic or manual. And final will be the curtaining machine, that is the packing into the drug products. Next one is the suspension. For suspension preparation, there is number of booths. For example, dispensing booth, colloidal mill, which will be attached with the manufacturing tank, storage tank, bottle washer, and dryer. And last is the emulsion. So for emulsion preparations, there will be the Silverson mixer emulser, filling and sealing machine, labeling machine, and automatic filling machine. Now we will go for the semi-solid department. In semi-solid department, so there are four types of packing is there. First one is the size reduction, that is a colloidal mill, emulsifying and mixing machine, filling and sealing machine, and labeling machines. Now we will go for the solid department, which is the heart of the production planning. So mostly depending upon the area and depending upon the volume, there are different types of solid departments. So for that, first one is the tablet. For manufacturing of the tablet, machineries are to be used. Okay. So there will be same dispensing, size reduction, mixing, double cone blender, and granulation. In granulations, there are two types of machines. Wet granulation, that is the high shear granulator, that is the FBD, and dry granulation, that is the roll compactor. In capsules, there are two types of capsules, hard capsule and soft gelatin capsules. In parental preparations, there are following machines to be used, that is the ampoule washing machine, sterilization of the ampoules, liquid preparation tank, filtration membrane tank, and storage tanks. Next one is the aerosol and the spray products. So in the preparation of aerosols, there is the manufacturing of the product concentrate, that is the active ingredients plus propellant, addition of the propellants and other additives like vehicles, antioxidant and surfactants. Next one is the R&D department and QC and QA department. So these are the departments which has to be considered during the layouts. Coming to the next, as we know that we have completed about the pharmaceutical production management plannings. Now we will go for the technology. Overall, the exposure of mine is working in a solid oral development. So I will give the brief informations what the technologies are going on and what are the engineering point of view, because theoretical part of point of view we are very well known to the tablets, coating, dispensing, fluidizations. These are the terms which are used to, but actually what the commercial point of view it's going through, we will come to know. So coming to the next slide, that is the scaling up. So the area of interest is my technology transfer. Before going for the R&D scale up, so in R&D development, that is the small development is there. So laboratory point of model. Second one is the scale up model, pilot model, and last one is the production model. So as we can see that 1, 2, 3. So in this scaling up means what? We have to scale up the batch in a such a way that we have to meet the market volume. For that reasons, whatever the capacity that RMG must having, so it should be complies with the occupancy as well as the batch size. Okay, so this is the scaling up. So for that, we will go for this step by step. What is the pharmaceutical product development? Go to the next slide, ma'am. Okay, so initiation of the project. So first we will go for the design of experiment, that is the initiation of the project. In that we can define the factors in which, that is the CMA and CPP. So CMA is nothing but the excipient, that is the raw material starting initial, that is the critical material attributes and what will be impacted on the critical process parameter, that is the CPP. So we will analysis for the risk analysis of the both. On that we will select a design, that is the select the proper experiment design which will affect the factors, that is the critical process parameter. Now coming to the major responses, that is the CQA. So I will just explain what is the function of the CMA, CPP and CQA. CMA is nothing but the critical material attributes, that is the starting material of the excipients which will impact the my process, that is the procedure, and whatever the final products which will be coming, that is the CQ, that is the critical quality attributes. So these are the factors to be considered well during the initiation of the product development. So these are the design of experiment to be run in experimental runs. So analysis factors that includes the response relationships. So first will be the model graphs. in which we can apply the numerical factors and graphical analysis. Then we will decide the range, on what range the product should be coming. So range is nothing but the giving a proper range which indicates that the process will be feasible, and it will indicate that critical capability factor. So now the design DSV is nothing but a design space verifications, in which the design has to be done in a such a way that the product will meet the parameter, and the product final product which will obtain, which will be in line with the specifications. So once the CSV has been controlled, that is the control design verification has been done, the design of project will be ended, and the space will be in a control way. The design space is nothing but a knowledge which is obtained during the initiation of the project R&D scale. So this is nothing but the pharmaceutical product development at the start of the project. Now we will go for the next slide, in which we have defined what is TPP and QTPP. So starting with the target product profile and quality to target profile. So it will show the low impact to the high impact. If it is the high impact, we have to reduce the impact in a low way. So this can be obtained by considering the factors. So we will go for the QBD project start. So first one is the profile of RLD, that is the innovator sample, in which we can do the reverse engineering, in which the analysis of the RLD is nothing but a reference list drug, which is the innovator sample. So by comparing the innovator sample in a shape, size, average weight, okay, the coating of the tablet, the excipients, what the pill is giving. Pill is nothing but the patient information leaflet. So whatever the drug summary, that is the summary of the product characteristics, has been given in a FDA site, we will compare that product, and we will develop replicate that product in a generic form. So this is the nothing but the profile of RLD, that is the drug product. Once the QTPP has been identified, so we will identify the quality attributes. So in identifying the risk factor, the criticality analysis, and finally we will obtain the CQA, that is the critical quality attributes. Once the design of experiments has been satisfied, so we will determine the ideal region of the quality. And depending on the model analysis, we will define the CMA and the CPP. Okay, so once the design of space verification has been controlled, the establishment of the limits has been done. So we will go for the manufacturing requirement analysis, and for that management the product life cycle, and we will go for the ending of the project. So this is nothing but the QBD project ends. So this is the initiation of the project, looking after the small scale up in the R&D and converting into that commercial point of view on a high scale. So this is nothing but the project review. Coming to the next slide. Now, as a technology part of view, so would like to introduce what are the contents I am going to explain regarding the RMG. RMG is nothing but the rapid mixer granulator. Second will be the FBD, that is the fluidized bed dryer and processor. Multi-mill, quadruple-mill, roll compactor, blender, compression machine, and autocoater. So these are the contents which are going to be highlighted, and some of the parameters from the engineering point of view, how the scale activity has been done without prior going for the commercialization. So first we will go for the RMG, that is the high shear granulations. So you can see that the pictorial representations. Okay, so there is, from the left side on the green is the CMA, and the right side is the CPP. So CMA, what will be the CMA? That is the critical material attributes. So whatever the ingredients which are being used for the development of the product, that is diluent, okay, binder. So these are the intragranular material. So for that we have to decide what kind of binder concentration we are going to prepare. What is the granulation solution agent, that the concentration of solution to be added. Second one is the API diluent ratio. How much concentration of the API that is present in the formula. And we should know what is the API PSD, particle size distribution. What is BD, that is the bulk density and tab density. So why we are highlighting the PSD, BD and TD? Because these are the three factors which will useful for calculation of the RMG capacity. Why capacity is we are highlighting so much? Because once the capacity has been identified, we can manufacture the drug substances into the drug product. So why we are using binder concentration, granulating agent, API diluent ratio? These are the factors which will be impacting the critical process parameter. Why? Because if the concentration of binder is very high, there is a chances of granulating agent, means the granules which we have to be prepared will be of high density, and that high density of granules which will be impact by the CQA, that is the critical quality attributes. Why I'm saying that CQA? Because high granulating agent, high density of granules, high density of granules, high dt means the tablet formation will be of high dt. High dt means high distributed time. Once the dt time is retarded, my dissolution will be retarded. So these are the factors had to be considered during formulations, during the scale up activity. Okay, so that is only we are highlighting the CMA, CPP, and CQA. So this is only the binder concentration in the CPA, and the CPP will the binder spraying and addition rate. So why we are saying the binder spraying and additional rate? It has to be controlled, because if the time is given in sufficient manner, so we can add the binder in a suitable way. If time is not mentioned, because time is a variable, if we are not concentrating of time and we can add the binder within a second, there are chances of lump formations, and that will impact on the granulating chopper rate. Okay, so there are two types of machines, means we can say that designs in RMG. One is chopper, one is impeller. Impeller is a ground flow and chopper is the side one tangentially. So the impeller role is to form a slump. Lump formation will be done with the help of impeller, and chopper will, what will the chopper will do? It will break down the lumps in a appropriate size. So this breaking and formation of lumps will enhance the density of the granular weight mass. So this is nothing but the high shear granulation. Coming to the next slide, FBD, that is the fluid bed granulations. Here also I am highlighting the CMA and CPP and CQA. So in fluidized bed granulations is the next step of RMG. Once the weight mass is being developed in RMG, we will go for the drying. So drying includes that how much air volume is required to fluidize the weight mass. Okay, so as you can see that the API binder polymer concentrations. Now in that fluidized bed granulations, there are two types of granulation can be done. One is the drying of the weight mass and second one is the top spray. So I have highlighted that the API binder polymer concentration in a top spray manner. If the concentration of API and binder polymer is high, so the viscosity will be high and the weight mass will become the overloaded. So the weight mass cannot be fluidized due to the choking of the dish mesh. So depending upon the CMA, this will impact my CPP. That CPP is nothing but the whatever the temperature I require, that is the outlet temperature, inlet temperature, filter bag pore size. So you are unknown to that filter bag pore size, because filter bag are nothing but these are the bags which are inverted into the higher side and which are used to shake the materials and to prevent the clogging of the filter bag pores. So that's why we need to shake the filter bag during the operation measure. So next one is the number of spraying heads, binder spraying rate, pressure, gun to bed distance. Why it is said that a bed to gun distance? If the bed to gun distance is very close, then there are chances of overmass or weight formation. If proper gun to bed distance is being maintained, so the granules will be done in a fine way and the proper fluidization will be take place. For that reasons, we have to optimize the gun to bed distance also. And last one is the product temperature. So bowl capacity, inlet temperature, and fluidization air velocity. These all factors are representing to the CPP. Now, if the CMA is proper, there will be less CPP, means critical process parameter will be less, and that will not impact the CQA, because the CPP is being controlled. So the CQA will be in a controlled way only. So whatever the final in process blend which we are required, that will be the granules, PSD, BDTD, and the flow property. So the in process blend is okay, then the finished product, that will the tablet, CU, assay, impurity, moisture content, and dissolution will be in a proper way. So this is the engineering point of view where we have to work for the fluidized bed granulation by doing the calculations. Okay. Coming to the next slide is the tableting process. Okay. So now after the first one is the RMG, second one is the fluidized bed. So wet mass is converted into the granules. Once the granules formation has been done, so I have skipped one thing, that is the blending. So in blending, the blending is having a CMA. CPP is the critical process parameter is the blending time and the run, that is the RPM. So I have just skipped, but the main thing is the tableting process. So I will just explain the time tableting process. So the CMA and CPP are correlated. Once the particle size which has been to be controlled during the fluidizations in FBD, so we will have an impact on the tableting machines. So I will just explain what are the CMA to be considered during the formulation of tablet by using the machines. So the particles and granule size, that is the distributions and the shapes and the size. Second one is the fines ratio. So once the tablet blend has been completed, there is a ratio has been finalized during the scale up development. So I am saying about this fines and ratio, what it means. So fines and oversized granules means they should be the fines of 60% and 40% should be the ratio of granular material. Why the ratio has been decided 60 as to 40 like that? Because once the tablet has been formed, the blend will be moved in a dye cavity. Okay. So coming to the rheology point of view, so to prevent the void space, whatever the void space that will be having granular, and the void space which is being emptified, so that the void space should be filled by the fines. So once the void space of that granular material has been filled by the fines, the finished product of the tablet will have a good appearance. That is only we are deciding for the fines and oversize ratio. Okay. So for that reasons, we have to calculate the BD and TD, that is the bulk density and tab density. Once the fines ratio has been finalized, so we will go for the hardness, brittleness, plasticity, and elasticity. So the moisture content is also one of the part, that is the CMA. Why it has been considered as a CMA? Moisture content means what? If the blend having a high rate of moisture, it will impact my hardness. The tablet will withstand the hardness, that is the tensile. Means it will be form a tablet but have does not have any hardness that will impact my coating procedure. So for that reason, moisture content to be controlled during the blend formation in tableting process. So these are my CMA. Now we will go for the CPP. What is the critical process during the tableting process? So ejection force. What it means by ejection force? Ejection force is nothing but the tablet when get compressed in a unit mass, it has to be ejected for the tube channel. For that, ejection force is required. If the ejection force is not proper, the tablet will break in a die and it will be scrap out with a broken shape. For that reason, we have to control for the ejection force also. Why it is given this compression force? Second one is the compression force. The compression force is a very critical process parameter because the compression force which the roller has been giving now, in that the compression will withstand with a high density, means high load of the compressed force will be applied to the tablet formations and it will impact my in process physical parameters of the tablet. We will get the hardness, but the DT and dissolutions will be retarded for the future purpose. For that reasons, compression force to be optimized. Now we will go for the tooling design and dimension and scoring quality. So in the pictorial diagram, where we can see that tableting process flow, how it formations, upper punch and lower punch is there, cam filling is there, the tablet blend will come into the die fill and it will move, the die will be getted, the upper punch and lower punch will form a tablet in a unit mass. Okay, so once the unit mass is formed, the tablet will be ejected and it will come down from the ejection chute. So these are the overall symptomatic form for the tablet formations. Okay, now we will go for the tablet dies, access of material, powder volume control, hopper design. Last one is the hopper design. Why we are saying that hopper design is to be selected during the formulation? Because depending upon the in-process blend characteristic, if the hopper design is not appropriate or proper, there may be chances of formation of rat holing due to the fines and oversize ratios. We might be we can say that we are very convenient or we are used to the word for the rat holing formations. Technically the rat formation is very worst case because it will be impacted my cu, average weight and hardness and dt and all the things for the future purpose. For that reasons, hopper design is also a part of that cpp. Oh, okay. Next one we will go for the end calculation. Next slide. Yes. For that also we have the CMS and CPP. So the overall capsule formations, just a practical point of view, we can just elaborate how the capsules are being coming. They are getting separated in a cap shape and body shape has been separated. Once the capsules has been separated, then it will come to the blend where there is slug formation will take place. Depending upon the different pins are there, tamping pins. So five types of pins will be there. So one by one the slug will be formed in a different way, and once the weight has been selected, the slug will come into the cap shape, and next step will be the ejection of the means closing of the caps and the body, and final will be of the faulty. So this is the capsulation process. Now we will include the CMA. In the CMA, the cell material empty, the caps and body dimension. Yes, it is also very important part because the dimension and the capsule body shape has to be come to the inland with the channels. Once the channels is appropriate with the size and shape, there is a free flowing movement. If the dimension is different, there will be choking of the tablets and it will impact the further process. Okay. Next one is the gelatin bloom stem. The tablet capsule should have the bloom strength, means what? It had a suitable strength that should withstand the ejection force or the volume or vacuum which has to be used during the separation process. Now we will go for the fill material, that is the PSD, BD, TD flow. Percent of fines is also important here. Now the CPP is will be the powder bed height. So this totally depend upon the practical approach. What do you mean by powder bed height? So the blend which is to be come in contact with the capsule machines has to be suitable height. Okay, so tamping force is nothing but the formation of the slug in a different size. So once the slug formation has been done, so tamping will take place. So depend upon the number of tamps, how much the tamping is done for formation of the slug, it will depend, and dosing the speed. So why dosing this? Dosing this is nothing but it is the disk calculation is being done depending upon the size of the capsules. For example, there are different types of capsules as we know theoretically 000, 1, 2, 3, 4. Okay, so depending upon the thickness, volume of the capsules, the dosing disk selection has been done. For zero capsule, as per the calculations, there will be 4.5 thickness of the dosing disc will be calculated. So these are the overall brief information. Technically, you can come in contact when further there will be a technically debate will be there. So these are the CMA and CPP regarding for the encapsulations. If you are controlling the CMS and CPP, what are the impact of the CQ will be the capsule appearance, locking length. Why we are saying that locking length? If the capsule size 0 we are using for the capsule filling, the locking length will be the 17. If the slug formation is not proper, its overfilling is there, the capsule length will be enhanced. Once the capsule length will be enhanced, the packing issue will be replicated. So for that reason, locking length is very important for the CQA. Now we will go for the next slide, that is the roll compact. Yes. So roll compactors. So R&D scale up, commercial scale up, we are using that roll compactor, the drug substances which are very sensitive for the moisture, sensitive to the organic solvent. For that reasons, we can skip that API by using the wet granulations or drying process. For that reasons, we are going for the roll compactor where the flow property can be enhanced by giving the pressure to the API as well as the excipient to form a proper granulations. Okay. For that, the CMA is API excipient ratio. API excipient, again the same thing, PSD, BD, TD, flow properties, API particle size, and polymorphisms. Why we are saying that polymorphs? Because most of the molecules are there. When it is coming into the compression force, that roller compression has been taken place, it might be changes polymorphic change. Okay, so polymorphic change will be done. So for that reasons, we have to first only we have to go for the CMS, and the last one is the dry binder and lubricant concentration. These are the CMS which are impact my CPP. So CPP. So just I will explain what are the roll compactors machines and what are the what we can say that the machinery and terminologies are there. So first one is the agitator. Agitator, we can say that for the flow of the movement from the one end to another end, for that reason we are using the agitator speed. Second one is the feeder. Feeder means what? To move for the particular volume of the blend for the forward step or for a forward movement, for that we are using a feeding. Proper feeding will be there, proper agitation will be there, so they are proportionally constant. Okay, means they are equivalent or proportional to both of them. Then we are coming for the roller gap design. Why we are using the roller gap? Once the roller gap width has been optimized, so there will be formation of the ribbons. Ribbons is nothing but the converting the coarse molecule into the solid form. These ribbons are having a high density. So these ribbons will be passing through the milling stage, where the milling of the ribbons will be take place, and that improper flow materials will be converted into the flowable materials. For that reasons, roller pressure, roller speed, roller shape, surface design, and roller gap. These are the CPPs which has to be critically monitored during the roll compaction process. Okay, so granular milling speed is there, where the milling of the machines will take place. Sorry, milling of the blend will take place, and the fine ribbons will be converted into the granular space. Now, CQA, what will the CQA for that roll compactions? Ribbon appearance. Ribbon, why I am saying the ribbon? Because the roll compactor, the blend is converted into ribbon like threads. Means number or thread like structure will be there. It is having a good length. This will impact, means having the good strength to hold the granules. And these granules will be converted into the milling, that is the granulating milling speed. So for that, the CQA will be ribbon appearance, thickness, density, porosity, breaking force. And overall, there will be the granule PSD, BD, flow property, assay, and blend. So these are the engineering point of view where the technologies are coming, where we are converting the solid drug substances into drug products. So whatever the technology point of view we are moving theoretically, this is only the commercial point of view where we are facing number of issues from the selection of the excipient to the dissolution point of view. So these are the parameters, okay, in RMG, in FBD, in tablet processing, in encapsulations, or in roll compactions. These are the parameters we have to follow. And these parameters are nothing but the very critical parameters which will impact our dissolutions, or means whatever the finished product specifications. So for that reason, we are using this kind of technology in a commercial point of view. So theoretically, we have the concepts, but in practical point of view, the concepts are totally different. But the basic should be the same applications. That is only the my motto is that the technology what we are going through now to be delivered in a simple way. Okay. Now coming to the next slide, man. Okay. So this one is the scale up basics. Now, whatever the CMA, CPP for RMG, granulations, FBD, drying processes, for roll compactions, whatever designs we have gone through. So we have to go for the geometrical similarity, kinematic similarity, and dynamic similarity. So engineering point of view, I will explain what is my geometric similarity. The two systems are called geometric similar if they have the same ratio of the characteristic linear dimensions. Linear dimensions means what? The two cylindrical mixing vessels are geometrically similar if they have the same ratio of height to diameter. Means same height and same diameter means they can say they are geometrically similar. Now we will go for the kinematic similarity. Two geometric similar systems are called kinematic equivalent similar. They have the same ratio of velocity. Okay, same velocity, that is kinematic similarity. Same geometrical shape, geometrical similarity. Now we will go for the dynamic similarity. They are said dynamically similar when they have the same ratio of the forces. So geometric, kinematic for velocity, and dynamic forces. So these are the terminologies which are very useful for the compilation, for the scale up basic, for the going for the RMG, FBD, blender, and tableting force. So why the forces here, dynamic forces are there during, we can correlate now. For dynamic forces, we can say in tableting process, compression force is there. Okay, velocity. Why velocity? In FBD, there is the air velocity will be there. The volume of air is required to fluidize the blend of higher weight mass. For that reason, kinematic similarity is required. For geometric similar means same cylindrical size means RMG. RMG having a different size, different geometrical, but the height to the diameter of impeller and the diameter of the RMG will correlate to the RMG. Okay, so that is why we need to get the scale up basics. Coming to the next slide. So overall, we can have what are the factors to be considered when we are going for the different types of based on the occupancy of the equipments. So we have different of equipment for the RMG is there, mill, Cotroco mill is there, blender is there, fluidized bed dryer is there, compression machine is there, auto coater is there, and roll compactor. So this all I have highlighted for the CPP and CMA. Now before going for the selection of the occupancy as well as the equipment, so we have to consider some of the factors. So for RMG, we have to calculate the fill ratio, impeller strip speed, Newton power number for the torque. I will explain one by one for the next coming slides. So blender, Frod's number is there, number of rotation is there, headspace calculation is there. For fluidized bed dryer, there is a base plate area ratio and occupancy is there. For compression machine, dwell time, DT. And for auto cutter, brim volume for occupancy, pan load, and Frod's number for the pan speed. And roll compactions, roller gap, and roller forces. So these are the factors which has to be considered during selection of the equipments for going for the in process as well as the finished products. Okay. Coming to the next slide, RMG. So I will just explain in a short because it will enable means it will understand you, but it will be not implemented from your side. So I will just explain in a simple way. So fill ratio is nothing but the ratio of the bed height to the RMG bowl diameter. Okay. So in the first slide we have just mentioned the RMG. So whatever the height of the impeller and the bowl diameter will give the fill ratio. Okay. So this is nothing but the why we are using this fill ratio to check the occupancy of the RMG to have the capacity to withstand that blend. For that reason, we are calculating the fill ratio. Okay. Coming to the next slide, we will go for the kinematic similarity. So RMG can be scaled down or scaled up according to the tip speed. Why tip speed is required? Because from the R&D, small scale up unit is will there. That is a 20, 10 liter, 20 liter, 5, 50 liter. But commercial point of view, there will be 100, 200, 400, 600, 1200, 1400. So to match the tip speed of R&D or to match the tip speed of the commercial, we have to calculate the tip speed. So for that tip speed, we have to calculate the formula is given 2 pi Rn divided by 60. So R is nothing but the radius of the RMG and the N is the number of impeller. Okay. So radius of the impeller and the number of RPM and divided by 60 is nothing but the we are calculating tip speed in a meter per second. So the tip speed represents the linear velocity at the tips of the impeller. Okay. Coming to the next slide. So we will go for the dynamic similarity, that is the force. So Newton power number can be used to scale the torque. So formula for the NEP is equal to P divided by density R raised to 5 N raised to 3. So P is nothing the power consumption by the impeller blade. Torque is nothing but the torque into 2 into pi into D N. So N is number of the impeller RPM, R is the impeller radius, and density P is nothing but the wet mass density of the granules. So this is Newton power number relates to the drag force acting on a unit area of the impeller and the inertial strength. So when we will come to the know what I am completely I am saying that what is the chopper. So it will come when it will be when you will be the used to with that terminologies. So impeller and chopper are nothing but they are used to formation and breakdown of the lumps. So whatever the drag force which will be acting on the impeller and inertial strength will indicate a Newton's power number. Okay. Coming to the next slide, RMG's relative swept volume. So relative swept volume is the volume of the material that is to be swept per second per volume of the bowl. So the bowl is nothing but the RMG. This parameter is calculated as the volume swept by the blades per unit time versus the bowl capacity. It is related to the work or energy imparted on the wet mass during the granulation. So impeller swept volume is nothing but the impeller height divided by bed height divided by 100. So we will get the RSP volume. So coming to the next slide, that is the fluidized drying, top spray and bottom spray drying process. So on the next slide, LBP booster calculation will be there. Yes. So if the fill ratio of the equipment remains same in all the scale, then in any scale the air velocity should be remain same. Why we are saying the velocity and capacity, the blend? Because to withstand that blend weight mass, we require the velocity to fluidize the weight mass. For that reason, the air velocity foot per minute is calculated. So its formula is nothing but the air velocity is equal to air flow, that is cubic foot per meter, divided by the base plate area. Now, coming to the first slide, you will see that in the previous slide, yes. So first slide, so on the right side on the corner of the top, there is a base plate. Okay, this base plate, it indicates a size of a sieve of a mesh. Okay, so for 100 mesh, 200 mesh, so this mesh should be have a proper fluid, means air volume will be passing through this and there will be proper fluidization. If that mesh is getting choked, the air volume will be decreases and the fluidization of the material will not take place. For that reason, we have calculating the base plate area. Okay, for the base plate area, next slide, ma'am. So in that area, we have just given an example that what the factor is to be calculated. So in R&D, there are the machines having the capacity of 125 cm square. Means it is indicate that its capacity is 125 liter. And now we will go for the commercial 1300 liter. So whatever the factors we are going to use, so whatever the air volume for a scale A and scale B, so spray rate for a scale B is equal to Q1 into A divided by to A1. So Q1 is nothing but the spray rate of scale A, batch gram per minute. A2 is the base plate area of the scale 2, and A1 is the base plate area at a scale 1. So now we are talking about the scale 1, scale 2. This will just for your information. When you are coming to similar with that terminologies, so you will use this scale 1, scale 2 means whatever the factors we have to calculate for the commercial point of view, we will use that scale 1 and scale 2. For calculating of the spray rate, from R&D to the commercial, we are using scale 1, scale 2 means whatever the base scale area of the base plate of commercial and base plate of area of that R&D, we will consider that factors, and on based on that we will calculate that what is the factor to be calculated for going for commercialization on a higher level. Okay, so this is only the basic information. Coming to the next slide is the blender. Okay, yes, in pictorial representations, there are two types of blender have been shown. One is the plan view and one is the evaluation view. So the plan view means what? The blender, how it is rotated, means considering the height, head space, what we can say that. And next one is the stable portion, elevation view. So blender mixing and PLB process. Blender is nothing but it is a process where the mixing of the blend is being carried out. Next slide. Okay, so mixing, and as we are familiar with the terminologies lubrications, where the glidants and what anti-adherent materials are been added in a last stage, and lubrication to be done for the materials for the proper flow, proper movement of the blend. For that reason, we are using the blender. So while going for the blending from R&D scale up to the commercial point of view, so we have to calculate the factors. So first one, we will go for the geometrical similarity, keeping the ratio of all the lengths constant. That is the percent occupancy. And dynamic similarity is nothing but the maintaining the constant forces. That is the Frod's number. So we will calculate the Frod's number is nothing but omega squared into R divided by G, where R, the omega is nothing but the rotational speed. R is the IBC radius, that is the rotational length divided by 2. We will get the radius. And G is the gravitation constant. So this formula will give the Frod's number. Means in R&D, what Frodd number is there to match that R&D scale up, we have to calculate for the commercial scale up. Okay, for that reason we are calculating the Frodd's number. So here is the example given, because in blending there are two main variables are there. One is mixing time and one is blending time and mixing RPM. So blending RPM is nothing but how much RPM is required to mix blend for a particular time. For that, two variables, time and RPM. So in R&D scale up, time is fixed. So commercially, we have to go to add to calculate the factor to replicate that tip speed as well as the number of revolutions for a commercial level. We have the example is given. So omega 1 into R1 is equal to omega 2 into R2. So omega 2 we have to calculate. So omega 2 into R1 divided by R2 raised to half. So by calculating that, we will calculate that what RPM is required for the commercial point of view. In R&D, there is a 10 rpm for 12 minutes. For commercial, for 5 minutes. For how 5 rpm, how much minute is required? So the time of high scale blender is nothing but the 120 divided by 5 is equal to 24 minutes. Means for R&D, we are calculating for 12 minutes. Okay. For higher level, we calculate for 24 minutes. So these are nothing but the calculation factors which are used during the scale up activity. Okay. So we have given number of examples. You can get these examples and you can have these examples for the study purpose or for the use purpose. Coming to the next slide. So I have given numbers, sorry, examples. Coming to the next slide. Hello. Hello.
Yes, yes, Mr. Jayesh.
Sorry. Yeah. Coming to the next slide, ma'am.
Just a minute.
Yes.
Maybe. Uh,
Okay. Thank you.
is it okay?
Yes. Yes. Okay. So
Yes.
just I will just complete this all the activity. So first one slide there is example for the lubrications. You can get these examples and you can calculate the how the number of revolutions can be calculated from the small scale to higher scale. Okay, and what is the time required to be calculated from the R&D to the commercial point of view. Now we will go for the compressions. So in that there are two variables have been shown in the in the pictorial, that is the PCD, that is the pitch circle diameter. So this pitch circle diameter is nothing but which is used to calculate for the dwell time. Dwell time is nothing but what is the time required for the tablet formations when upper punch and lower punch will come in contact with both each others. So different types of punches are there, D type and B type. So D type having the head flat is 15.875 mm, that is 0.625 inches, and the B type punches head flat will be 9.525 mm, that is a 0.375 inches. Coming to the next slide. So compression machine speed determination is based on the del time, as I have explained. So del time formulation formula is the PHF, that is the punch head flat into the 60,000 divided by pi into PCD into n number of rotations. So this is the formula which is used to calculate the dwell time from small scale equipment to the higher scale equipment. Okay, the example is given in next example, next slide. For okay, so in a R&D small scale, that is CTX 28 machine is there and 51 station is there. So from small scale, how to calculate the higher rate? So based on that factors, we can calculate that whatever time RPM is required to match the speed of the higher machines. So these are the basic examples are given. Now we will go for the next plate, that is the determination of the fill depth. Fill depth is nothing but the how much powder blend is been filled in a depth capacity. It depend upon the size of the can. So the maintaining the fill depth, we can achieve the desired tablet weight, thickness and hardness of in any scale. So fill depth formula is nothing but 4m divided by pi into d square into d. So the tablet diameter, that is the length and density of the mass, is used for the formulation. So now last stage is the coating. Next slide. So while selection of the coating machines, we have to consider the brim volume. So brim volume is nothing but the total volume of the water when the pan is fully filled and its current positions. So as we know, Acela quota, normal quota pan is there. So in academic point of view, we are very used to with the Acela quota is there. Number of coating pans are there. But for commercial point of view in industry, there are different pans are there. That is the different mix pans are there. We can see the palm plan is there, clad pan. So different capacity, that is the 36 inch, 15 inch, 26 inch, 24 inch, 660 inch, 66 inch. So depending upon the different size and capacity of the coating pan, we have to calculate the brim volume. So brim volume is nothing but the volume of the cylindrical part plus volume of the conical part. Okay, so for example, we are given the brim volume of 60 inch Gansen coating pan is having the capacity of 497 8 liters, and the BD of tablet is 0.67 gram per meter for 100% occupancy. So we will calculate that 497.8 into BD, we will get 100% occupancy is equivalent to 3352 kg. Means volume is in liter, we will divide multiply by density, so little bit get cancelled and the mass will come. So that's why the most of the capacity of the coating pen will be in the liter form only. We will multiply it by density, so we will get the volume will get converted into the mass. For that reason we are using the brim volume. So the batch size is 280 kg and the occupancy would be 280 divided by 3352 into 100, we will get the occupancy is 83.95. So this occupancy, why it is required? Because to finalize the batch size, if the volume is more and the capacity is less, so it is not possible to coat the tablets. For that reason, the capacity of and the volume of the volume for the coating machines is required. For that reason, scale-up factor is very important. Okay. So these are the next slide is the pan coating. So for that reason, spray rate and how the inlet airflow is to be calculated is given in that. So spray rate for the large scale is 7.6 into spray rate of small scale, and airflow 7.16 into airflow of small scale. So these are the only terminologies which are used for the calculation of the pan load from small scale to the higher scale. So the formula is that pan load of large scale batch divided by the pan load of small scale batch into the pan diameter of small scale batch divided by the pan diameter of the large scale batch. So these factors will become when you are come to used to with that factorial designs. Okay. So next one is same example is given, pan speed, how to calculate the pan speed. Next slide. Okay, roll compactions. Next slide. So in roll compactions, we have to calculate for roller gap and roller diameters. For example is given, if roller hydraulic pressure is used for the conversion factor between the roller hydraulic bar to roller force K1 to be applied. So there are two types of machines. I mean, commercial point of view, WP 120 means the capacity of 120 kg, and WP 200, that is 200 indicates the 200 kg capacity. For WP 120, the kilonewton bar is 0.092. For W200, 0.698. So while calculating, we will get the roller force pressure, roller force or roller pressure. Okay. So this is only that slightly representations. So now last one is that one keynote is been given. A smart man makes a mistake, learns from it, and never makes that mistake again. But a wise man finds a smart man and learns from him how to avoid the mistake altogether. So these are the all representations regarding the scalar factor calculation based on the occupancy as well as the engineering point of view, and what are the factors which have to be considered while calculating the batch size in technology. So overall, I have just conjugated the both pharmaceutical production management as well as the technology which are going through commercially and we are used to. So this is the brief introductions and whatever the slides we have presented. Any questions regarding them? Just free to ask. Hello.
Yes, any questions?
Actually, just I will interrupt you, ma'am. Sorry for that.
Yes, yes,
Yes, the students who are very unknown to this process, I accept, I agree for that. But these are the scenario of the current situations, because you are the new aspirants which are going to be introduced in this technology, and we will definitely help for that. For that reasons, while coming to introduce for the new surprises for you, just for that reasons I have given you the detailing about the factors, what has to be calculated, what has to be considered during the scale up of R&D projects, and from going to the R&D projects to the commercialization for the larger volume. That's why I have just taken the selective points to calculate the factors. Just to have a brief idea, you can mail me or you can ask me regarding all the things. So this is only the brief introduction. Detailing is not there. You will come to know when you will be similar used to with the terminologies. That's what you will get little bit diverted with the topic, but it's very useful when you will come to day-to-day life. You will come and implement in that factors, so it will be really helpful for all of you. So that is only my concern. So sorry for that. The terms will be a little bit different for you, but it's a reality for the coming scenario. Okay, ma'am.
yes, yes, yes. You very good presentation you have done. Thank you, Mr. Jayesh. You have explained each and every aspect of the pharmaceutical production management with all technologies uh,
Yes, ma'am.
for all the dosage forms
Hmm.
and for different pharmaceutical formulations, which students are really wanted to know, right?
Yes,
So
yes,
I
yes.
would like to thank, and I have means students are having some of the questions. I receive it, but because of the time constraint, I'm going to
Yes,
share these questions with you, and later on we'll have the discussion with the students also. As you have told, I'm going to share your
yes,
email ID with them, right?
yes, yes, definitely, definitely.
Yeah. So
Yes,
with this, I would like to thank you, Mr. Jayesh, for the
yes, yes,
nice information and sharing your knowledge with our students and all our
yes,
participants. I would also like to thank Parul University, management of Parul University, and our Dean Faculty of Pharmacy, Dr. Abhay Dharamsi sir, to give us this platform to make this
yes,
kind of interaction with our students and faculty members. I would like to thank our PU webinar cell as well as our PAP webinar coordinator, Dr. Asha Patel madam, for cooperating always. And at last, but not the least, I would like to thank all the participants to join us.
yes, ma'am.
Thank you, everyone. Thank you,
Yes,
sir.
ma'am. Yes, yes. Thank you, ma'am. Yes, have a good
Sir.
day. Thank you for all
Yeah,
the
good.
supports. Okay, have a good day.
Thank you, sir.
Yes, ma'am.